the next thing we're going to focus on is RNA. So like DNA, RNA is a type of nucleic acid that is found within our cells as well as bacterial cells. There are some core differences that I want you to understand between DNA and RNA. One of the first and most important differences is the fact that while DNA is a double-stranded molecule, which we can see here, RNA is actually a single-stranded molecule. Now, it's still composed of the same nucleotides that compose DNA. It's just that DNA is a double-stranded molecule composed of nucleotides. So again, we have two single strands of nucleotides that are bound together by hydrogen bonds between the nitrogenous bases, the A's, the T's, the G's, and the C's, in order to get the double-stranded DNA molecule. Over here with RNA, RNA is simply just a single strand of RNA nucleotides. So we need to talk a little bit more about these nucleotides because there are similarities, but there's definitely some differences. So if we look at an RNA nucleotides still have a phosphate group, so that's no different. However, the sugar is different. If we look back, DNA had a 5-carbon sugar known as deoxyribose. RNA nucleotides still have a 5-carbon sugar, but it is a ribose sugar, so the actual sugar is different. And then the third and final difference has to do with the nitrogenous bases. If we think back to DNA, each nucleotide has one of the 4 nitrogenous bases, A, T, G, or C. It's a little different with RNA. In RNA, the nitrogenous bases can still be A, can still be G, and can still be C. However, there is no thymine in RNA. Instead, we replace thymine with uracil. So in RNA, your nitrogenous base options are A, U, G, or C. So there are similarities, definitely, but to focus on those differences, DNA is a double-stranded molecule composed of nucleotides. RNA is a single-stranded molecule composed of nucleotides. DNA is going to we're going to have the deoxyribose sugar within the nucleotides, whereas RNA is going to have the ribose sugar within the nucleotides. And then lastly, RNA is going to have uracil as a nitrogenous base, whereas DNA was going to have thymine as a nitrogenous base, and they can both have adenine, guanine, and cytosine. There are different types of RNA. So the 3 different types of RNA that we'll talk about in this course are rRNA, which is ribosomal RNA, and this is used to form the ribosome, which is involved in protein synthesis. And then there's mRNA, which is messenger RNA, which is what is produced after transcription and contains the code for how to synthesize a protein by the ribosome. And then lastly, there's tRNA, or transfer RNA, which is used to transfer amino acids from the cytoplasm to the ribosome during translation. So the first kind of process we're going to get into is replication. We'll be focusing on replication and then ultimately transcription and translation. But to start off, replication is the process of duplicating DNA. So you are going to take, one DNA chromosome and you're going to make 2 DNA chromosomes. And this is what bacteria typically do when they're ready to go through a binary fission cycle. Ultimately, they can then take DNA and turn it into RNA, which is a process that's done during transcription. And then finally, that RNA can be translated during translation into protein. But first, we'll start out with replication. So basically, replication is duplicating DNA. So if we have one double-stranded DNA molecule, what we do is we open up that double-stranded DNA molecule into 2 single strands, and then we start to make a copy of each one of those single strands. So the original strands here in the blue, and the new copies that we're making are here in this orangish yellow. So that's being done more advanced here, And then ultimately you get 2 double-stranded DNA molecules. So there are a series of enzymes that we're going to talk about that make this happen, but just to kind of step back and get an overall view, you are taking one chromosome and you're opening it up into 2 single strands of DNA. You're making a copy of each one of those single strands to attach to the original strands, and you're ultimately ending in 2 chromosomes. One that will stay with the original parent cell, and the other is going to go to the new daughter bacterial cell. When all cells replicate their DNA, they do so in what we call a semi-conservative pattern, which basically means part old and part new. So if we look right here, we have a double-stranded DNA molecule that is actively going through replication. So it's being ripped open into 2 single strands, which is what we see if you just look at the blue. The blue is the original parent strand. So that's the original strand that was opened up. And then we make a copy of each one of those strands, which the new strand is here in the red. And in the end, you get 2 chromosomes. But you can see each chromosome is part old, which is the blue, and part new, which is the red. You don't get one that's entirely an old chromosome and one that's entirely a new chromosome. They are made in a semi-conservative pattern where they're part old and part new. So one strand always comes from the original DNA strand, whereas the other strand has been newly synthesized. So in the next portion, what we're going to do is we're going to talk about replication, kind of looking at specific enzymes and kind of drawing it out in a diagram so that we can learn all about how DNA is replicated in bacterial cells.